Okay, children. So today I am going to discuss experiment number 5, children, in ninth class. So see here, experiment number 5. What is experiment number 5, children? To study the onion peel cell and temporary amount of onion peel cell and cheek cell, children. Okay, here we will see studying of onion cell and cheek cell. Children, onion cell comes under plant cell. Cheek cell comes under
Animal cell.
animal cell. What are the major difference between plant cell and animal cell, children? Plant cell have
Vacuole.
cell
Cell.
wall and large vacuole and plastids. Many differences these are. And animal cell don't have cell wall. Animal cell don't have cell wall and they have small vacuole and they don't have plastids. They don't have plastids. Okay, children. Here cell wall is made up of what, children? Cellulose.
Cellulose.
Cell wall is made up of cellulose. Okay, children. So these differences we will see, children, here. Okay, uh, in the onion cell and cheek cell, children. For here, for you seeing onion cell, we use saffranin, children. Saffranin. For onion cell, we will use saffranin, children. Okay, so for onion peel, and for this one we will use methylene blue. Methylene blue. These are the stains, children, to color the cells on the slides. Saffronin is a stain used for plant cell. Methylene blue is a stain used for cheek cell, children, for coloring the cells to see under the microscope. Okay, children. So what is the procedure? Okay, so see here, children. Slowly, I will say the procedure for onion cell. Please listen, children. Here, first you take a onion using forceps to remove the peel, children. Remove the peel using forceps. Remove the peel of the onion, children. Okay, with the help of needle, take one needle, children. Using needle, separate the small portion of epidermis. Small portion of epidermis. Using needle, small portion of epidermis you have to remove, children. Small portion of epidermis. Now what you do? You will take a watch glass, children. You know watch glass. You know watch glass. In this we will put some saffron in, children. In this we will put saffron in watch glass, we'll put saffron in, children. Put this peel which you have taken to the needle, children, put this small peel in this watch glass with brush and allow it to stain for 3 to 5 minutes, children. This peel we'll put here, children. This epidermis peel is there, no, children. Here we'll put and we'll keep it here for 3 to 5 minutes, children, so that it will absorb the stain, children. It will absorb the stain. Okay. So after keeping for 3 to 5 minutes, transfer the stain peel to under watch glass that contains water in it to remove the extra stain, children. Now again, here the same peel will put in the water, children. We'll put in the water. Why will put in the water? To remove extra stain. To remove extra stain. Okay, same this epidermal peel after 3 to 4 minutes we'll keep it in this, no, children, it will absorb. To remove the extra one, we'll keep in the watch glass containing water, children. Now what you will do, transfer the stain peel with needle and brush onto the middle of the slide, the peel. Okay, children, you take a you take a slide, in the middle of this you put here, children. Gently you put. Okay, now what happens, on we have to keep slowly, children. After keeping here, remove, children, here a glycerine you have to put, children. Glycerine. Why do you put glycerine? They may ask one more question. Why do you put glycerine? To avoid drying of the peel, children. To avoid the drying of the peel. Try to understand, children. Here, after putting here, we'll put glycerine, children. On this epidermal peel, we'll put glycerine. Why do you put glycerine? To avoid drying. Now on this after putting glycerine drop, now on this peel, children, on this epidermal peel, put cover slip. Cover slip. I hope you all know cover. What is cover slip? A small mirror-like thing will be there, children. That cover slip we will put. After putting cover slip, children, put filter paper. Put filter paper on that and absorb extra glycerine. Absorb extra glycerine. And remember, children, while putting cover slip, see that no air bubbles are formed inside. If air bubbles are formed inside, you can't see the under the microscope. Now, children, after removing the extra stain, clean the slides of the cover slip and start seeing under the microscope. When you see under the microscope, when you see under microscope, what you will see, children? Because this is an onion peel, what you will see under the microscope? It is a plant cell, children. It is a plant cell. Nicely you can see a cell wall. Okay, you can see number one cell wall. You can see nucleus. You can see big vacuole. Big vacuole. How you will see means nucleus will be at certain corner, big vacuole will be there, children. Okay, and lightly stained cytoplasm is present in the cell. Okay, children, always remember saffranin should be dilute, children. You should not take very concentrated one. Means how to make it dilute by adding some water. Avoid the formation of air bubbles when putting the cover slip. Okay, and take thin pill, children. Don't take very thick pill, you can't see. And use clean and dry slide. Wipe out extra stain of water present on the sides of the slide using the filter paper, children. These are the precautions that we are going to take, children. I hope you understood very easy one, children. One thing, first you will take peel, you will put 3 to 5 minutes in saffronin. Then you will put in water to remove the extra stain. Then you will put on the watch glass. Then you will put glycerine 1 drop. Why will you put glycerine, children? To avoid drying of peel. To avoid drying of peel. Why you put glycerine? To avoid drying of peel. Then on this you put filter paper without air bubbles. And extra glycerin, whatever it is there, use filter paper and remove it, children. Okay, and when you are putting this cover slip, see that no air bubble comes here. Now see under the microscope, you can see cell wall, you can see nucleus, you can see big vacuole because it is a plant cell. Hope you understood, children. No rocket science is involved. Very easy one. Nothing is there. Okay. So children, now we will see cheek cell, human cheek cell. How to take human cheek cell? Okay, temporary mount of human cheek cell, children. This is experiment number 5, children. Very important. Previously, in previous days, used to have MCQs, children. And nowadays, they have decreased the number of MCQs, but they are asking children. See here. So, how we will take? Same, children. So, take like this one will be there. No, children. Our toothpick will be there. No, children. Toothpick. Take toothpick. Okay, from the side of your mouth inside. Okay, just slowly you scratch, children. Don't hurt yourself. Slowly you scratch, and in the watch glass you put methylene blue, children. In the watch glass you put methylene blue. In the watch glass you put methylene blue, children. Okay. Now what you will do? So what you will do, children? The scrap, what you will take from the side of the mouth, put in this, children. That scrap. Okay. Here also same, children. Just for 2 to 3 minutes you put like this on the, children. Okay. After that, same, keep in the watch glass to remove extra methylene blue. Take a slide, children. On the slide you put here this, children, that human cheek cells what we have kept. Same, children, put glycerine again. Same glycerine. Again on this you put cover slip, children. Whenever you are putting cover slip, see that no bubbles will form, no air bubbles will form, and remove extra stain using filter paper. Remove extra stain by filter paper, children. Same thing, children. Filter paper or blotting paper, whatever it is. Filter paper or blotting paper, remove the extra stain. See under the microscope. Same procedure. Only difference is here we are using methylene blue. There we are using saffronin. Okay. So same, children. Now what you will see under the microscope? What you will see under the microscope? Irregular shapes are seen, children. Irregular shaped cell we will see. Irregular shaped cell we will see. No cell wall, children. No cell wall. And normal nucleus, small nucleus only will be there, children. Here, small nucleus only. Sorry, small vacuole will be there, and small nucleus will be there, children. Small nucleus and small vacuole, children. Because here, here in plant cell, vacuole was very big and nucleus is small. But here, normal nucleus will be there, normal vacuoles will be there, children. So what are the precautions you should take, children? When you are scrapping, children, unused toothpick you take, children. Don't use used one. Okay. Placing of unused means once you are using this, put in the dustbin. Next time I will do laboratory experiment again. I will use that only. Don't use like that, children. Okay. And this, when you are putting cover slip, children, see that no air bubbles come inside. That is very important. Avoid overstaying also. Don't put it for 5-10 minutes. Only 2 to 3 minutes is enough, children. And remove the extra water using blotting paper. Avoid overlapping of the cells, children. You have to see under the microscope, children. Under the microscope, you will see normal vacuoles and normal nucleus, children. This is how it is done, children. So I hope you all understood very easy. Here also same. Here also same. In watch glass, you are putting in the dye. Then you are putting in the water to extract it. Here also after putting the cells, you are putting glycerine. Glycerine is to avoid the drying of the cell. Glycerine is to avoid the drying of the cell. Glycerine is to keep the cell moist to avoid drying. Then you will put the cover slip. When we are putting cover slip, you will see that no air bubbles are formed, children. when no air bubbles are formed, children, now you will see at the microscope. When you are seeing under the microscope, here in plants you will see very big vacuoles, here small vacuoles you will see, children, and here cell wall will be there, here no cell wall, because plant cells don't have cell wall. Sorry, plant cells have cell wall, animal cells don't have cell wall. In plant cells, vacuoles are big, in animal cells, vacuoles are small. In plant cell, plastics are present, in animal cells, plastics are not present, means chloroplast is not present. Here will stain with saffranin, here will stain with methylphenidate. Here plasma membrane is present. Plasma membrane is made up of lipoproteins. Plasma membrane is made up of lipoproteins, lipids and proteins. Lipids means fats, proteins means proteins. Okay. Who help in for, formation of plasma membrane, children? Which cell organelle? Anyone? Which cell organelle helps in?
Endoplasmic reticulum.
Very good,
Endoplasm.
very good. Membrane biogenesis. Do you remember, children? I told membrane biogenesis. Who will do? Smooth or rougher?
So both, sir.
No, mainly smooth. Mainly smooth. Mainly. Rough endoplasmic reticulum have ribosomes. No, none. Ribosomes helps in preparation of proteins.
Protein.
Rough endoplasmic reticulum has ribosomes. Ribosomes helps in preparation of proteins. Smooth will prepare fats, means lipids. These lipids and the proteins together, membrane biogenesis occurs in smooth endoplasmic reticulum. Children, you have to remember, membrane biogenesis is done by endoplasmic reticulum. Endoplasmic reticulum helps in the formation of plasma membrane. Plasma membrane is present, cell membrane is present in both onion cells and cheek cells. Okay, they are made up of lipoproteins, means lipids and proteins, means fats and proteins. Whereas cell wall is made up of cellulose. Cell wall is made up of cellulose. Okay, children. So this is the fifth experiment, children. I hope you all understood. Very, very important. Please, I will tell.